Hello everyone and welcome to our channel MooTechie. It's so wonderful to have you joining us for another exciting session. Today, we're venturing into the fascinating microscopic world to explore the fundamentals of bacterial genetics. This is where we unlock the secrets of how these tiny organisms store, use and share their genetic information. It's a truly incredible topic, so let's get started. We'll be covering several key areas in our lecture today. We'll look at DNA replication, the regulation of genes, how genetic information can change through mutation, and the fascinating ways bacteria exchange genes. We'll also touch on the amazing world of genetic engineering in medicine and its application to clinical diagnosis. It's a full agenda, so let's dive in. First, let's talk about DNA as the genetic material. Inside a bacterial cell, the genetic information is stored in DNA, which is organized into a single circular chromosome. This chromosome is found in a region of the cell that is free of ribosomes. The replication of this DNA happens bidirectionally, and remarkably, the entire process can be completed in about 40 minutes. In contrast, smaller genetic elements called plasmids often replicate unidirectionally. Now let's visualize the structure of the bacterial chromosome. Imagine a circular chromosome which is like a twisted ladder, the famous double helix. Each side of this ladder is a strand made of a sugar-phosphate backbone. The rungs of the ladder are pairs of nitrogenous bases held together by hydrogen bonds. The four bases we find are adenine, which pairs with thymine, and cytosine, which pairs with guanine. This specific pairing is the very key to genetic inheritance. Next up, let's discuss plasmids. Plasmids are extrachromosomal genetic elements. This means they are small circular pieces of DNA that exist independently of the main chromosome and are autonomously replicating. Almost all are circular, with an exception like the one found in Borrelia burgdorferi. Crucially, plasmids do not encode functions essential for basic survival, but they provide additional genetic information that can give the bacterium a special advantage. These advantages can include antibiotic resistance, the ability to produce toxins or bacteriocins, and special metabolizing properties. Plasmids come in different types. Large plasmids like the fertility factor F or resistance transfer factors can actually mediate their own transfer from one cell to another through a process called conjugation. Smaller plasmids are often not conjugative, meaning they don't encode the proteins needed for transfer, so they are more sedentary and don't move between cells easily. We'll explore the main methods of gene exchange: conjugation, transduction, and incorporation a little later. Now let's look at a diagram illustrating bacterial chromosome replication. We start with a parent chromosome, with its two parent strands. As replication begins, an enzyme complex moves along the DNA. New nucleotides, each consisting of a deoxyribose sugar, a phosphate, and a base, adenine, cytosine, guanine, or thymine, are added to the growing strands, with hydrogen bonds forming between the base pairs. The replication process moves in a specific direction, and on one strand we see the formation of short segments called Okazaki fragments. Finally, we end up with two fully replicated chromosomes, each containing one original strand and one newly synthesized strand. It's a beautiful and precise process. This brings us to the central dogma of molecular biology, the replication of DNA, which is then followed by transcription, and finally, translation into functional proteins. Let's visualize the first part of this process: protein synthesis, specifically transcription. Inside the cell, we have the chromosome made of DNA nucleotides. Each DNA nucleotide has deoxyribose, a phosphate, and one of four bases, A, G, C, or T. The enzyme RNA polymerase binds to the DNA and builds a strand of RNA. RNA nucleotides are slightly different. They have a ribose sugar and the base uracil or U instead of thymine. This process creates a single-stranded RNA molecule. There are three main types of RNA produced: messenger RNA or mRNA, which carries the genetic code; ribosomal RNA or T component of ribosomes; and transfer RNA or tRNA, which brings amino acids to the ribosome. Now let's see the next stage in this diagram: protein synthesis through translation. First, the messenger RNA or mRNA moves from the chromosome to the site of protein synthesis. In the larger subunit of the ribosome, the genetic code is read. A specific amino acid is activated by an enzyme called synthetase. This amino acid is then attached to its corresponding transfer RNA, or tRNA. Each tRNA has a set of three nucleotides called an anticodon, which recognizes and binds to a complementary codon on the mRNA strand. As the ribosome moves along the mRNA, bonds form between the incoming amino acids, building a polypeptide chain. This all happens with the help of the small ribosomal subunit, and the process continues, reading the codons on the mRNA, which are made up of the bases A, G, C, and U, until a complete protein is synthesized. Now, how does a cell control all of this? Through the regulation of gene expression. A cell must constantly adapt to changing conditions, and it uses elementary regulatory mechanisms to do this. This helps minimize energy requirements, turning genes on only when they are needed, and off when they are not. Often, genes for enzymes in the same metabolic pathway are grouped together in a unit called an operon. An operon includes a promoter, the genes themselves, and a terminator. This allows them to be coordinately regulated, transcribed, and translated as a single unit. There are two main types of transcriptional regulation. The first is negative control, where genes are expressed unless they are switched off by a repressor protein. The second is positive control, where genes will not be transcribed unless an active regulator protein called an AP inducer is present. Operons themselves can be classified. Inducible operons are switched on when a specific substrate is introduced, leading to the expression of enzymes necessary for its metabolism. Repressible operons are normally on, but the presence of the end product of the pathway reduces the amount of enzymes produced. Genetic information isn't static, it can change. Damage to DNA can lead to a mutation, which is an accidental change in the sequence. Fortunately, cells have DNA repair systems to fix these errors. Bacteria can also acquire new genetic information through the exchange of genes menace, a process called recombination. So what is a mutation? A mutation is any change in the base sequence of DNA. A single base mutation can be an insertion, a deletion, or a substitution. Remember, DNA is transcribed by RNA polymerase into mRNA, which is then translated by tRNA. The tRNA carries specific amino acids that recognize a set of 3 nucleotides, a codon, on the mRNA. Any change in the DNA sequence can alter the codon, leading to a different amino acid being incorporated into the protein produced by the ribosomes. Mutations can arise in different ways. They can occur spontaneously during replication. They can also be induced by external factors. Heat can cause deamination. Ultraviolet light can cause pyrimidine dimer formation. Ionizing radiation can cause the opening of the ring structure in the bases. Chemical mutagens, such as nucleotide base analogues that mimic real bases, frameshift mutagens that cause insertions or deletions, and DNA reactive chemicals that modify the base structure, can all induce mutations. Thankfully, cells have several repair mechanisms for DNA. Direct DNA repair involves the enzymatic removal of the damage. Excision repair involves cutting out the damaged DNA segment and synthesizing a new one to replace it. Recombinational repair retrieves missing information by using a sister chromosome as a template. The SOS response is activated when replication is interrupted, but it can be error-prone, sometimes leading to more mutations. One of the most exciting aspects of bacterial genetics is gene exchange between bacterial cells. There are 3 main mechanisms. First, transformation, which is the acquisition and incorporation of exogenous or foreign DNA from the environment. Second, transduction, where DNA is transferred from one bacterium to another by a virus called a bacteriophage. And third, conjugation, a quasi-sexual exchange requiring direct cell-to-cell contact. Bacteria frequently exchange DNA, which can then be integrated into their chromosome or maintained in plasmids and passed on to daughter cells. Let's explore transformation in more detail. This was famously demonstrated by Griffith's experiment with colonies of encapsulated and non-encapsulated Streptococcus pneumoniae. Transformation is the ability of a cell to take up and stably maintain exogenous DNA. Some bacteria have natural competence, an ability to interact with exogenous DNA. This is not a permanent feature, and often occurs toward the end of logarithmic growth. Examples include Haemophilus influenzae, Streptococcus pneumoniae, and Bacillus species. In the lab, we can induce competence using chemical methods or electroporation. Now let's look at a diagram of bacterial transformation. In Griffith's experiment, we see 1. A live S strain, which is encapsulated and virulent. 2. A live R strain, which is non-encapsulated and non-virulent. 3. A heat-killed S strain. 4. When a mixture of the live R strain and the heat-killed S strain is injected into a mouse, the mouse dies, and live S strain bacteria can be recovered. The process shows a chromosome or DNA segment from the donor being taken up by a recipient bacterium across its cell membrane. The dividing bacterium then inherits this new DNA, which can confer new properties like the capsule. Next, let's discuss conjugation. This process involves direct cell-to-cell interaction and is unidirectional, from a male donor to a female recipient. There are different conjugative plasmids. The fertility factor, or F factor cell, allows an F plus cell to make contact with an F minus cell using a specialized pilus. This leads to a wall-to-wall contact and the formation of a cytoplasmic bridge. This contact initiates replication and transfer of the plasmid. Antibiotic-resistant plasmids, or R factors, can also be transferred this way. In gram-positive bacteria, an adhesion on the surface of the donor mediates the contact. The following page, titled Conjugation, visually represents this process of conjugation. Let's examine this diagram on bacterial conjugation. It shows two main types of transfer. On the left we have plasmid transfer. An F plus donor bacterium, which contains the F factor plasmid, makes contact with an F minus recipient bacterium. The plasmid is replicated, and a single strand is transferred through a conjugation pilus. Once inside the recipient, it is made double-stranded, resulting in a new F plus daughter cell. On the right we see chromosome transfer. This can happen when the F factor has integrated into the donor's chromosome, creating an HFR or high-frequency recombination strain. During conjugation, a part of the chromosomal DNA can be transferred along with the plasmid DNA, leading to the formation of a recombinant F plus bacterium. Another major method of gene exchange is transduction, which is mediated by a bacteriophage. A bacteriophage is a parasitic virus that infects bacterial cells, using their energy and protein synthesizing machinery. It can contain DNA or RNA as its genetic material. During infection, only the nucleic acid enters the bacterial cell. Let's look at the structure of a typical bacteriophage in this diagram. We see a complex structure with a head or capsid which contains the genetic material. Below that is a contractile sheath surrounding the tail. At the bottom, we find a base plate and tail fibers arraign, which help the phage attach to the specific receptors on the surface of a bacterial cell. Bacteriophages have different life cycles. The first is the lytic cycle, where the phage takes over the cell's machinery, replicates itself, and ultimately causes lysis or bursting of the cell. Phages that follow this cycle are called virulent phages. The other life cycle is the lysogenic cycle. In this cycle, the phage does not immediately lyse the cell. Instead, the phage DNA is integrated into the bacterial chromosome. A phage that does this is called a temperate phage. It can remain dormant like this for many generations. Under certain conditions, it can be induced to enter the lytic cycle. Sometimes, the integrated phage DNA can also convert the bacterium, giving it new properties. This diagram helps illustrate bacterial transduction. On the left, we see generalized transduction. During the lytic cycle of a phage, a random piece of bacterial DNA from the donor bacterium can be mistakenly packaged into a new phage particle. When this phage infects a new recipient bacterium, it injects this bacterial DNA, which can then recombine into the recipient's chromosome. On the right, we see specialized transduction. This involves a temperate phage that has integrated into a specific site on the donor bacterium's chromosome. When it is excised, it sometimes takes a small adjacent piece of bacterial DNA with it. When this phage infects a new host, it transfers this specific set of genes. Now let's talk about mobile genetic elements called transposons. Transposons are segments of DNA that can move or jump from one position to another in the genome, or even between a chromosome and a plasmid. There are different types: insertion sequences, which carry only the genetic information needed for their own transfer; complex transposons, which carry additional genes, like those for antibiotic resistance, and are often part of our plasmids; and phage-associated transposons. This ability to move genes around leads us to the incredible field of genetic engineering in medicine. Scientists have developed vectors or vehicles that allow the cloning of any DNA sequence. This means that eukaryotic genes, like human genes, can be expressed in prokaryotic systems like bacteria. Since many genetic diseases are caused by the lack of a specific protein, we can use bacteria as factories to produce these proteins for replacement therapy. This is also how we produce recombinant vaccines, like the one for hepatitis B. There's even research into using engineered bacteria for replacement therapy, a concept known as bacterial interference. Let's see a simple diagram of the genetic engineering process. We start with the donor plasmid and a piece of foreign DNA that we want to clone. A restriction enzyme cuts both the plasmid and the foreign DNA at specific sites, creating compatible ends. The enzyme DNA ligase then joins the foreign DNA fragment into the plasmid, creating a recombinant DNA molecule. This recombinant plasmid is then introduced into a bacterium, where it replicates independently from the chromosome, producing many copies of the gene of interest. Finally, let's look at how this knowledge is applied through molecular technologies in diagnosis. A key method is the use of nucleic acid or DNA probes to diagnose and study diseases. In this technique, a DNA sequence of interest is inserted into a bacterium, amplified to high copy numbers, and labeled. This labeled probe can then be used to detect complementary DNA sequences in a patient sample through a process called in situ hybridization. Another revolutionary technology is the polymerase chain reaction, or PCR. This technique allows for the generation of millions of copies of a specific piece of nucleic acid from a suspected microorganism, making it incredibly easy to detect very small amounts of a pathogen. And that brings us to the end of our whirlwind tour of bacterial genetics. From how they replicate their DNA and control their genes, to how they evolve and share traits, and finally, how we harness this knowledge for medicine and diagnostics. It's a field that truly powers modern biotechnology. Thank you so much for your curiosity and for joining us on this exploration of the microscopic world. Thank you. See you in the next class.